evening. So today our guest speaker is Dr. Lucy Godley from Chicago, United States of America. The topic is identifying potential germline variants from sequencing hematopoietic malignancy. It will be promoted by Mumbai Hematology Group, and NATCO Oncology is a sponsor of this program. And the Perfect Square will take care of all the logistics for the meeting from the start to end. So before starting, just a little introduction. I want to thank you, Mr. James Rajkumar and Team NATCO for supporting this beautiful meeting. Mr. Yash Kalpesh and the Team Perfect Square for the logistics. Executive Committee of Mumbai Hematology Group. Obviously, our guest speaker, Dr. Lucy Godley from Chicago, United States. All our discussions who are themselves eminent hematologists or medical oncologists, and you who are participating for sparing your Saturday evening. So thanking all. So tomorrow there will be another lecture around 11:30 a.m. Spreadsheet to bedside, unfolding cost effectiveness studies for a clinician by Dr. Joseph John from Department of Clinical Hematology, Christian Medical College, Ludhiana. Today is our discussions are spread across the India from Dr. Aditya Jandial from Chandigarh, Dr. Amit Khurana from Surat, Dr. Harsha Prasad from Bangalore, Jasmita Das from Delhi, Dr. Karthik from Chennai, Dr. Mahadeva Sami from Goa, Dr. Mukul Agarwal from Delhi, Dr. Preeti Mehta from Mumbai, Dr. Soumya Mukherjee from Calcutta, Dr. Seta Lunkar from Pune, and Dr. Tufan Dolai from Calcutta. So to that, Aditya is a hematologist from the Department of Clinical Hematology and Medical Oncology, Postgraduate Institute of Medical Education and Research, Chandigarh. Dr. Amit is a hematologist, hemato-oncologist, and BMT physician from Surat, and very close friend of mine. Dr. Harsha Prasad is a consultant pediatric hematologist and oncologist, assistant professor, Department of Pediatrics, Kasturba Medical College Hospital, Manipal, Bangalore. Dr. Jasmita Das, associate professor in hematopathology, Department of Hematology, All India Institute of Medical Sciences, New Delhi. Dr. Karthik is a hematopathologist, associate professor, Department of Oncopathology from Cancer Institute, Adyar, in Chennai, far south of our country. Dr. Mahadeva Swami, again, the western part of our country, from the Goa. He's a hematologist and bone marrow transplant physician from Manipal. Dr. Mukul Agarwal, assistant professor, Department of Hematology, All India Institute of Medical Science, New Delhi. Dr. Preeti Mehta, she is a pediatric hemato-oncologist from HN Reliance Foundation Hospital, Saifi and Raheja Hospital, Holy Family, from Mumbai. Dr. Soumya Mukherjee, consultant hematologist and hemato-oncologist from Apollo Hospital, Kolkata. Dr. Seta is a consultant hematologist and BMT physician from Pune. Dr. Tufan Doloi, hematologist from Calcutta. So now Dr. Lucy will deliver the lecture. She is a Hospira Foundation Professor of Medicine and Human Genetics, Section of Hematology Oncology, Department of Medicine, the University of Chicago Comprehensive Cancer Center, Chicago, United States. She had a special interest in the molecular basis of bone marrow malignancies and is an active researcher in the field. In her laboratory, she studies the basis for cancer cells' abnormal pattern of DNA methylation, as well as inherited forms of bone marrow cancers. She has received numerous awards for her research, including the Howard Hughes Medical Institute Physician Postdoctoral Award, the Cancer and Leukemia Group B CalGB Foundation Clinical Research Award, the American Society of Clinical Oncology Young Investigator Award, Clinical Research Foundation Young Investigator Award, the SWEP Foundation Cancer Development Award, and the Kimmel Scholar Award. She was inducted into the American Society of Clinical Investigations in 2012. Her goal is to improve health through a deeper understanding and appreciation of science by integrating knowledge about fundamental networks with the cancer cells and bringing novel insights into the pathophysiology of our patient's disease while offering them new therapeutic options. So now the lecture is identifying potential germline variants from sequencing hematological malignancies. Dr. Lucy, now I'm just stopping here. Now stage is yours.
Well, thank you so much. I really want to thank you, Dr. Agarwal, and all the other organizers for including me, and especially for all the participants to spend, as you said, Saturday evening with us. It's morning here for me, so it's not quite the same, but I really appreciate people spending that time. So let me share my screen. Here we are. And let me go to the slideshow. So hopefully now you can see the slides.
Yeah, we can see the slides.
Perfect. So I was asked to speak about finding germline DNA variants when you sequence a malignancy. And we will talk about that over the next hour or so, but I'm going to throw in some science as well. So hopefully that's okay for people. And I always like to begin by thanking people because none of this work could be done without any of these groups. First, on the left, you see a thank you note that the daughter of one of my patients gave to me, which is thanking me for taking care of her mom. It's supposed to be a picture of me doing a bone marrow biopsy on her mom, which is really sweet for me. But really, I want to thank the patients because it was really my interaction with the patients that made me feel that many of them were programmed to get their malignancies. And when I was coming through my fellowship, I was taught that this was exceedingly rare. But as I saw the patients and heard their personal and family histories, I was more and more convinced that there was a germline component. And so they really led me along the way to something that was underappreciated in the field. So my first thank you is, of course, to the patients and their families. They have been very, very active participants in this work. And then in our lab, we have a lot of very, very dedicated people who are really focused on this work. And so I really want to thank them for all of their dedication. On the clinical side, I really want to thank Soma Das, who runs our genetic services lab, which is a clinical laboratory in our Department of Human Genetics. And when I talked to Soma and Zeshwan Lee, who was her technical director at that time, about this underappreciation of germline predisposition, they were very willing to put together a testing pipeline, which has evolved over time, and we can talk about that. And they've been wonderful, wonderful partners in diagnosing our patients. Jeremy Siegel is a pathologist, and he runs the tumor-based profiling that we'll talk about. And Jeremy has been very open to using those data with my lens, so to speak, to see which patients would be at high risk for having those variants as germline variants. And finally, Dr. Vardaman is a wonderful hematopathologist who's now retired. But he and I discussed or argued over so many cases that he decided he really needed to advocate having germline predisposition within the classification scheme for myeloid malignancies at least. And I'm very happy that the World Health Organization and others have included germline predisposition in their classification schemes and in their guidelines. And there is really nothing sacred about the myeloid leukemias. This germline predisposition crosses all aspects of heme malignancies. And so I'm really very indebted to Dr. Vardaman for starting that. So as I was just saying, germline predisposition to the myeloid malignancies, and I would really extend that to all hematopoietic malignancies, is, I'm very pleased to say now, I would say widely recognized. The WHO classification includes germline predisposition to myeloid malignancies, as you know, and the newer version will include this as a permanent category. With the suggestion that lymphoid malignancies are also very often germline determined. We have different clinical guidelines now that urge testing for germline predisposition, and these are extremely important because at least in the United States, these kind of guidelines dictate insurance coverage. And the more the guidelines call for this kind of testing, the more we can get the testing paid for. And then our hospital is very happy because they underwrite the testing less and less when the insurance companies pay for it. So the one issue with germline predisposition is we keep discovering more and more genes. And so every month or two, I have to add genes to this list. Right now, I put these genes into these different categories, which just helps me think about which gene predisposes to what type of cancer. On the left here, you see genes that right now are associated with myeloid malignancies. In the next column, genes that are associated with lymphoid malignancies or immunodeficiencies. And I do have a bias that any germline variant that predisposes to immunodeficiency will eventually be shown to give risk to lymphoid malignancy. In the next column, you see genes where deleterious variants give risk for either myeloid or lymphoid malignancy. And in the last column, genes that confer risk to both blood and solid tumors. There's a lot of overlap there. Actually, you could even argue that MBD4 here from the myeloid list really ought to be in this column here, because now we see an association with colon cancer, uveal melanoma, and other cancers. So there's constant shifting back and forth, and I can't even keep the slides up to date. Now, another way of kind of making this impression to people that this is a relatively common phenomenon is we have this riddle that goes, how much does a ton of feathers weigh? And it's sort of a riddle or a paradox because, of course, each feather is very light. But if you have many, many feathers, you get something heavy. And equivalently, each one of these disorders may be relatively rare, but there are so many of them that it becomes a common phenomenon. And so I've tried to fit all these feathers on one slide. It's very difficult because there are so many of them. And I've put some of these feathers down here in large size because just take bone marrow failure or dyskeratosis congenita, that's comprised of many, many, many genes. The more we study, the more we add genes. And so this becomes a harder and harder problem, so to speak. We have to be broader and broader in our testing to be able to diagnose our patients accurately. Now, people always ask me, well, how often does this really happen? You say it's common, but how common is it? So we tried to get some data. We started this study many years ago. We tried a few different ways of answering this question. I'll show you one set of data that's published, and I'll talk about one that's almost published. So we tried to start with a population where you get a reasonable percentage. We didn't want to start with all AML, for example, or all MDS, because we thought that we might get a very small percentage. We wanted to restrict this to a population where we might get a pretty reasonable percent germline. So we decided to restrict to young MDS. So why would a young person get myelodysplastic syndrome? Here in the United States, and I'm sure it's true in India and all of your countries, MDS is an older person's disease. You can see the incidence here really peaking in the older age range. So if you have somebody in the younger age range, there must be a reason that they're getting MDS. So we arbitrarily chose the ages of 18 to 40, just because I'm an adult oncologist. And you can see here that we collected samples from all over the world because it's relatively rare to have young MDS. People were very generous to give us these samples, and these were generally bone marrow aspirates. And what we did in this case was we grew the mesenchymal stromal cells out of the bone marrow aspirate, because most of the cells in the bone marrow aspirate are going to be hematopoietic. They're going to be involved with the MDS. You're going to see somatic variants. So in order to find the germline variants, we grew the mesenchymal stromal cells, which are non-hematopoietic. They're from a different lineage. And that allowed us to call the germline. A little technicality for those of you who are thinking very carefully about it. People have shown acquired mutations in malignancy-associated mesenchymal stromal cells. So we demonstrated the same variant in the hematopoietic cells from these bone marrow mesenchymal stromal cells. So we had the same variant in two different lineages, and that's how we technically define germline. You can see here that most of our samples were from MDS. We did allow aplastic anemia just because of the diagnostic overlap with hypoplastic MDS. And the answer was about 20% of MDS was germline. Now, let me just say here, we called these variants very conservatively. So these are in genes. nobody would argue would predispose to myeloid malignancy. And these are variants nobody would argue are likely pathogenic or pathogenic variants. If we get looser and looser in our calling of variants, we allow variants of uncertain significance that are awfully suspicious. If we allow genes that we think probably should be on this list, this number goes even higher. We can get this number up to 80% if we get looser and looser. But we tried to be very, very strict, very conservative. No one would argue these calls. It's 20%. So this means it's at least 20%. And I would say to you, even likely higher than that. For aplastic anemia, we get 15%, again, very conservatively called. Now, unbeknownst to us, the pediatricians were doing exactly the same experiment. And what did they find? They found the same thing, germline predisposition in young people with MDS. And what's very interesting is the gene that's involved changes as the age is diagnosed. So if L, as you get into the teenage, young adult years, got a 2. As you get into the really, I would say, true adult years, we're talking DNA repair and telomere biology. And then we know that in the average age, we see DDX41. So yes, a 72-year-old and 85-year-old can have a germline predisposition. I think this is very interesting. It tells us that the age of presentation is just a surrogate for the pathway. This makes biological sense, right? If you have a germline mutation in something that gives you disease very quickly, you'll present younger. If it takes a little bit more time, you'll present in the teenage or young adult or young adult age group. Now you see here we have an age gap. We have a knowledge gap here in the 40 to 75-year-old. We're trying to fill that gap. We're trying to fill that gap 2 ways. The first way is repeating the same experiment, just using MDS diagnosed within that age group. And the second experiment that we did was using samples from the CIBMTR, the International Bone Marrow Transplant Registry, where they had peripheral blood from the MDS patient and the related donor. And in this case, we called germline variants because the variant was shared between the patient and his or her donor. Now, this will underestimate the true germline rate because sometimes those donors would be wild type and we can't call germline if the donor doesn't have it. Even so, with that underestimate, we get 7% of all MDS across the ages, germline. And where are those ages? Are they all confined to the young people or are they spread across the age range? The answer is they are spread across the age range. And again, the gene involved goes according to the age decile. So I think the CIBMTR work is, fingers crossed, almost accepted. So hopefully you'll see that very soon. And that will corroborate what we have here and start to fill in the gap. Now, as we study individuals and families with germline predisposition to hematopoietic malignancies, we keep seeing the DNA repair pathways, especially the pathway of homologous recombination. This is where we have DNA damage, especially double-strand breaks, being recognized by ATM. ATM phosphorylates CHEK2. CHEK2 then goes on to phosphorylate its downstream targets, P53, BRCA1, BRCA2, et cetera, that arrest the cell cycle and allow the cell to fix that DNA damage. If you have a defect in this pathway, you don't fix your DNA damage very well. You go on to get clonal hematopoiesis, especially with CHEK2 variants, and you go on to get hematopoietic malignancies. Now, we're very, very interested in this pathway because we keep seeing it and we keep trying to do more and more science about it. Just to give you some examples of what we see from the patients, on the left, you see a typical pedigree for what we see for germline CHEK2 mutations. The proband was a woman diagnosed with MDS in her mid-30s, and she went on to have an unrelated donor transplant. After transplant, went to have her breast MRI, had a very early breast cancer, had that taken care of, had her thyroid ultrasound, had thyroid cancer. And so it does feel a bit like whack-a-mole as these people survive. They get more and more cancers, but hopefully with good surveillance, we can keep treating them. Her mother had died of multiple myeloma, her uncle of a B-cell lymphoma. We know that both of those shared the CHEK2 variant. And so here for CHEK2, you see why we put this in the category of hematopoietic and solid tumors. And within hematopoietic malignancies, both myeloid and lymphoid malignancies. The variant that this family has is the I200T variant. Some of you may know it as the I157T variant. And it is the most common variant we see when we ascertain families on heme malignancies and look at the germline. You can see all the different variants that we see, but half the time we see this particular variant. Now, if you ask a breast cancer doctor about this variant, the breast cancer doctor will tell you, oh, that's a very mild risk factor for breast cancer. But as a doctor who sees blood cancers, I would tell you it's not so mild for the blood cancers because half of our families have exactly the same variant. This made me think that maybe the CHEK2 variants have a different risk in the different tissues. This variant is particularly deleterious in the hematopoietic space and less so in the breast or say in the colon or thyroid. And I think the issue here is what are the backup DNA repair pathways that are in place when you disrupt homologous recombination? I would say to you that the backup DNA repair pathways in the hematopoietic tissue are error prone. So if you try to fix that DNA damage with backup DNA repair pathways, you actually make the problem worse. Possibly in the breast and colon, you have more accurate backup DNA repair pathways. That would be our hypothesis right now. Now, for the I200T, one argument has been it's not really a cancer predisposing gene. You're only seeing it because it's relatively common in populations. And I have kind of a problem with that argument because I think the impact of a variant is only inversely proportional to its population frequency. So if you have a variant that's extremely rare, it probably has a strong effect, but a variant that's more common in the population can have an effect, it's just smaller. And so we did this burden calculation to ask, are we seeing the I200T as frequently as we're seeing it just because it's common in the population, or is it actually enriched in our patients? And we did this for the other S428P and the Del 1100C variant, which you probably are all familiar with. And in our population, we see statistical significance for the I200T, the one I just shared with you, also for this Ashkenazi Jewish founder mutation, S428P. In our population, we do not have significance for the Del 1100C. So this tells us that these two variants are enriched in our population. We think that they're really having a functional effect to cause the hematopoietic malignancy. Now let's just take a look at some of the sort of characteristics of the malignancies that we see. First, we asked, well, what kind of malignancies do we see in these patients with the CHEK2 variants? And here you can see that you can see both primary myeloid malignancies as well as therapy-related malignancies. You can also see solid tumors as you would expect. And you can see lymphoid malignancies as well, often, again, after the diagnosis of a solid tumor. And so we do think probably the chemotherapy and radiation that's being used for the primary solid tumor is damaging the bone marrow and contributing to the development of the hematopoietic malignancy. We've looked at the mutational spectrum in those myeloid malignancies that develop, and we really don't see a pattern of mutations. What we see is a pattern of chromosome changes. So we see statistical significance for balanced chromosomal rearrangements. This makes sense. if you have a double-strand DNA break and you cannot fix it very well, the cell will just stick those chromosome ends together and give you a translocation. And so I think it's not surprising that what we see is a signal in the cytogenetic abnormalities, not so much the molecular abnormalities. Now, as we were doing this work in the human, and as I'll show you in a minute, the mouse, this work from other groups came along. We were very excited about it. People are very interested in clonal hematopoiesis now. As you all know, the somatic mutation over time in the hematopoietic stem cell that gives its progeny out in the peripheral blood with an inflammatory milieu contributing to cardiovascular disease, hematopoietic malignancies, solid tumors now it looks like, COPD it looks like, lots and lots of conditions. So if you ask in the general population, are there germline variants that predispose to the development of clonal hematopoiesis? The answer is yes, check 2, and actually this exact allele, the I200T allele. So this makes us think that these particular individuals are predisposed to clonal hematopoiesis, and that's their stepping stone to getting their hematopoietic malignancy. I think part of the reason that these population studies were able to find this statistically is, again, this variant is relatively common in the population. And so you have the statistical power to be able to detect it. So this gave us a hint. And as I said, we were very curious to develop models that could convince people that this variant was having an impact in the hematopoietic space. So we thought, well, how are we going to prove to people that this particular variant makes a difference in the hematopoietic tissues? We thought, let's model it in the mouse. Now, I know a lot of people make very fancy mouse models. They have fancy promoters and they turn genes on and off in particular tissues. But people aren't like that, right? People either have the mutation or they don't. And so we decided to make an old-fashioned knock-in mouse, just a mouse that looked like the people. We made an old-fashioned knock-in mouse. So this mouse has the I161T, which is the equivalent position in the mouse CHEK2 gene. And we can breed it to homozygosity. Just like in people, you can have the I157T in the homozygous state. And we put those mice on the shelf and we asked, does this mouse get sick at all? And I have to say, in this regard, we were very benefited by the pandemic because most people for the mouse work keep the mice on the shelf for a year. And after a year, if they don't see anything, they generally start to sacrifice the colony because it costs a lot of money. And I have to say that year mark came about March 2020. And I was the only one in the lab because people weren't allowed to be there. And so I have to admit, I was focusing on the patients, not as much my mouse patients. And the mice stayed on the shelf longer than probably would have otherwise. So you can see now that the homozygous mice in blue, the heterozygous mice in red, they have a higher white cell count, have a higher lymphocyte count, a higher neutrophil count. And look what happens. They start to die around 17 months. And they really, really start to die by 24 months. What are they dying of? Are they dying of breast cancer, colon cancer, thyroid cancer, hematopoietic malignancies, or something else? Well, before we got there, we asked about the clonal hematopoiesis because we had that study from the human that first the humans were getting clonal hematopoiesis. And this comes from the impact panel that's developed at Memorial Sloan Kettering. And I really thank Ross Levine for helping us with this experiment. What we found was clonal hematopoiesis, actually in a dose-dependent way. So half of our mice, heterozygous, got clonal hematopoiesis and more of the homozygous. Now we have a much larger study that we've done, and we have the validation analysis cooking. So we'll really be able to say what percentage of the mice get clonal hematopoiesis, at what age, how do those clones grow over time. So I'm looking forward to those results. And what were they dying of? they were dying of hematopoietic malignancies. They get every type of hematopoietic malignancy you can name. T-cell leukemias, B-cell lymphomas, myeloid malignancies. They never get breast cancer. They never get colon cancer, never get thyroid cancer. So to the extent that a mouse can mimic a human, and obviously not perfectly, but the mouse would signal to us that this particular allele preferentially gets hematopoietic malignancies. Now you can imagine I really want to model the other Ashkenazi Jewish mutation as well, because you saw the odds ratio was even higher for that allele. So again, coming back to this pathway, we keep coming back to homologous recombination. We're very interested in ATM and different ATM variants, very interested in BRCA1. We were able to knock out BRCA1 in the mouse bone marrow and show that it's a Fanconi anemia gene. We've known that for BRCA2 for many years. Excuse me. So do pay a lot of attention to homologous recombination defects in your patients with hematopoietic malignancies. They are often germline. So now let's come back to the patients, testing and management. So who do we test? These are the current criteria that we use to test a person for germline predisposition. I give this about 5 years. I think we'll be testing everyone. Everyone diagnosed with a hematopoietic malignancy will have germline testing, just like they do flow cytometry, just like they do cytogenetics, molecular. Everybody going through transplant, all donors, related or unrelated. That's my prediction. We need to get there with data, but I think the data are coming quickly. Right now, we use these criteria. We ask about these criteria every single time someone has a bone marrow biopsy. Why at the time of the bone marrow biopsy? Because they're already having a procedure. Their skin is already sterilized for the procedure, and it's very easy to take a piece of skin. Why do we need the skin? Because in our case, for hematopoietic malignancies, there are floating, circulating tumor cells very often, and we don't want to mix up somatic with germline. So first of all, personal history, multiple cancers within the same person. We've talked about therapy-related leukemia for a long time, a therapy-related leukemia being one that arises after treatment for a solid tumor or some rheumatologic condition for which cytotoxic therapy is used. But actually, when we looked at our database, we had more patients who had survived a blood cancer, enough time to develop a solid tumor. We call those people double cancers. And actually, it doesn't matter the order of the solid versus the hematopoietic tumor. In either case, germline testing positivity rate is about 20%. If someone is diagnosed with a blood cancer at a much younger age, take young MDS, a much younger age compared to the average, there's something driving that, and we do germline testing. The caveat here is that I think people have assumed that if you present at an average age, you cannot have germline contribution. That is completely wrong. And so we think about germline predisposition in all patients regardless of age. And remember DDX41 for the myeloid leukemias, those patients present at the average age. We pay attention to family history. What do we consider a positive family history? Any heme abnormality or hematopoietic malignancy or young onset solid tumor within 2 generations of the proband. For us, young is less than 50 years old. This is an arbitrary cutoff. You could choose another age if you felt it was appropriate. And last, the topic of the discussion today, if we see a DNA variant that could be germline with a variant allele frequency consistent with germline in a gene known to confer germline susceptibility in tumor-based sequencing, then we consider looking to see if that variant is actually germline. These tumor-based tests are not designed to find the germline. We'll talk about why they are not comprehensive testing. But of course, if you have a germline variant, it's in all the person's cells, including their tumor cells. So if you happen to see it, you can't ignore it. And we'll talk about that. So this is the algorithm for our patient workup. Again, we take that personal and family history on everyone having a bone marrow biopsy, all acute leukemias. And I would say if our lymphoma doctors did more bone marrow biopsies, we would do more and more on lymphoma. Right now, we ascertain mostly on leukemia and myeloma because those are the programs that do the bone marrow biopsies. More and more, though, our lymphoma doctors are taking that personal and family history and referring their patients. Again, we look at that tumor-based profiling and look for alleles that could be germline. And then when we're going to do a related stem cell transplant, we really think about the family history. We do the skin biopsy, we grow the skin fibroblasts, and make the genomic DNA. Right now, our testing platform is an augmented whole exome. What do I mean by augmented? I mean that there are non-coding regions. These are usually promoters and enhancers where we know germline variants can exist, and they don't get covered very well or consistently with a whole exome platform. So we spike in extra probes that pull down those regions and add them to an exome. It's kind of a poor man's whole genome. For us, the genome sequencing is much, much more expensive than an augmented whole exome. So for the moment, we're comfortable with an augmented whole exome, but with time, I'm sure we'll go to whole genome. We do very special bioinformatic analysis to identify single nucleotide variants and copy number variants. Germline copy number variants are common. And again, these tumor-based assays are not designed for copy number variants. And so you have to be very, very careful to be sure that your testing platform can find these germline copy number variants. If we have a positive, then we do very extensive cascade testing throughout the family. If negative, then we can do a lot of research and discover new disorders, and we're working on maybe 4 or 5 of them now. So this is a constant effort. So let's talk about the tumor-based profiling again. When you have the germline, it is always going to be present. That variant will always be present about 50%, whether someone's in remission or not, because every cell of that body has that germline variant. Whereas a somatic variant, hopefully it's present when they have, the disease, but as they go into remission, that somatic variant goes down. Now, here's an example of where that really makes a difference. This is my patient. He was diagnosed with acute myeloid leukemia at 78. His mother had breast cancer at 52, smoker uncles who developed lung cancer. So none of the criteria fit to do germline testing on him right away. Think about using the somatic testing. So look at what happened with his somatic testing. At diagnosis, he had a DNMT3A mutation at 36% and a TP53 at 64. In remission, his DNMT3A went down to clonal hematopoiesis levels, as you would expect. The TP53 did not do that. It stayed about 50%. This man had Li-Fraumeni syndrome, germline TP53 mutation. You would never expect this from this family history. And I would say to you that every family we have identified ascertaining on heme malignancies, they do not fulfill any clinical criteria that have been defined for Li-Fraumeni. Why? Because Li-Fraumeni was defined with a certain characteristic complement of tumors. So people kept testing families that looked like that. So they were self-fulfilling prophecies, if you will. And this phenomenon that the clinical diagnosis, clinical definitions of these syndromes or disorders is too limited. It's true for all of these clinical descriptions. Now that we do molecular diagnostics, we see that the tumor profile is much broader than was originally defined. Now, if we take the tumor-based testing and plot the variant allele frequencies over time, we see this phenomenon. For TP53, mostly somatic, right? Those VAFs are all over the map. But a few times you can see people who have that VAF around 50%. Like the person I just showed you, these are 2 different families. So you can see how we can sort of pick up these families. Now, BRCA1 and 2, these are mostly germline. These are not often somatic mutations in hematopoietic malignancies. You can see sometimes they're somatic, but usually they're germline. So if you have BRCA1 and 2, on your tumor-based profiling and you see a variant, it's most of the time a germline variant. So can you look at the variant from the tumor profile and predict whether it's germline or somatic? For most genes, you cannot. The same exact allele could be somatic, could be germline. So we prioritize patients who have variant allele frequencies over, with our assay, 25%, or variants that are persistent over time, especially if the person has been in remission. 2 genes, though, we can look at the variants, and I would say with 100% accuracy, know whether they're germline or not. That's CHEK2. I've shared with you those variants, and DDX41. For DDX41, 99% of truncating variants are germline. Several mutations are common in particular population, the A500 frame shift in Asian populations, the D140 and M1 question mark variants in Northern European populations, and I'm sure there are others. The R525H is a common acquired mutation in the leukemia. So if you see the R525H in the context of a truncating variant, that truncating variant is, in our experience, always germline. Now, some people say, well, you're doing that tumor-based profiling anyway, just use it as your germline test. And you can't for the following reasons. First of all, I think we know which genes are mutated as somatic events in all heme malignancies at this point. And they are in exons. So that means you can develop a platform that's stable, just tile those exons and away you go. But as we've talked about, for the germline, we keep adding genes, and some of the variants are in non-coding regions. That means your platform has to change over time. That's why we went to the augmented whole exome, because if we have a non-coding region that gets identified, we just develop probes and add them in. It's really easy to expand the panel. If we have a new gene, we just pull down those variants from the exome and analyze them. We can go back in time and pull out the patients we missed. So the augmented whole exome gives us a lot of flexibility. It accommodates the change over time. We're not able to do that with a fixed platform. Biologically, the reason we should not be using somatic or tumor-based profiling is that's done on hematopoietic tissue. Hematopoietic tissue undergoes somatic reversion easily. That means if you sequence blood or bone marrow, you can get a false negative. This is why the families of SAMD9 and D9L variants were only identified in the past few years. Those families were identified decades ago, but people kept sequencing the blood and bone marrow, and there's a selective pressure in those diseases for correction of the defect in hematopoietic tissue. So the only way those families were found was finally when someone sequenced the skin fibroblast DNA. Again, we've talked about how important it is to be able to find copy number variants. You can do this with an exome panel or NGS panel with a microarray, or as we have done, going to an augmented whole exome and being very careful to use bioinformatics that can detect copy number variants. And lastly, again, we've talked about specific alleles. Most of the time, you cannot tell from your tumor-based profiling whether a variant is somatic or germline, but for DDX41 and CHEK2, you have a very, very good idea. We always validate with proper germline testing. And I should say, sometimes the way we do that is through segregation. So you don't always have to do skin fibroblasts. If you have family members and you see a DNA variant at a germline VAF in a relative, you know that it's germline. So sometimes we use family segregation to define germline. Now in the United States, we have lots of options of what testing platforms to use. As I told you, we developed a testing platform at the University of Chicago. Other commercial laboratories have done so, other academic laboratories have done so, but the coverage is all over the map. And whether that test can detect copy number variants or not is usually written in 9-point font at the bottom of the report. Most of our doctors who are busy in oncology practice don't have the time to read that fine print. And so I'm afraid a lot of people are really not getting comprehensive testing. I think the comprehensive testing is extremely important. What do you do if you find somebody with a germline allele? Well, what we do we look at the blood counts. We very often recommend people have a baseline bone marrow biopsy. For many of these conditions, there's baseline dysplasia. For all of the platelet thrombocytopenia associated disorders, ANCAR D26, ETP6, and RUNX1, for DDX41. So that's very, very important to have a baseline so that in time, in the future, if there are blood count changes, you do a bone marrow biopsy at that time, you have something to compare it to, and you can see if the dysplasia has changed over time. We follow people with very regular blood counts, potentially annual bone marrow biopsies, especially if the blood counts are changing. And then depending on the disorder, we may need to survey other organs. So for example, CEBP alpha germline mutations only predisposed to myeloid malignancies as far as we know. So we're fine with bone marrows and CBCs. Other conditions, say BRCA1 and 2, obviously just looking at the bone marrow is not sufficient. We have to be surveying other organs. So we give very specific cancer surveillance plans depending on the gene involved. Now, in the final few minutes, let's just talk about some clinical challenges. And these are actual quotes from emails that I've gotten to show you how difficult the work is. So patients and families believe in the germline more than at least my colleagues. Now, that's changing over time. But look at the date. That's not too long ago, right? The urgent need for germline testing prior to an allotransplant. We cannot do our testing quickly, but people keep thinking it's just like a blood count, right? And it's not. So this is coming from a colleague. I'm writing because you have a patient whose nephew is now under my care for CML. And I believe the sister, the patient's mom, is lined up to be the donor. And the mother reached out to us to inquire about whether germline testing was ever done. So here is the family pushing the doctors. Did you do that germline testing before you used my relative as the donor? The answer was no. The transplant team had not. But testing was eventually done, and there was a germline CHEK2 variant, and the donor had it. So the patients and the families think about what's in their DNA. They're pushing the doctors to do the right thing. Listen to our patients, right? That's always been the saying, and when my experience, very, very true. This is about a patient with a blood cancer who we asked the people doing the bone marrow biopsy to do that skin biopsy. And it was done on a day when the fellows were being trained. So the person doing the bone marrow biopsy was one of my colleagues supervising the fellow. And so we asked for the skin biopsy, and this was the email I got afterwards. Actually, you're probably aware of her family history, which sounds strange. The father had non-Hodgkin's lymphoma in the early 80s, not recognized until autopsy, involving the brain. Actually sounded like leptomeningeal tumor. Since that's lymphoid and she has an MPN, myeloproliferative neoplasm, I don't know that it's anything but coincidence, but interesting. So here's one of my colleagues not recognizing the relationship between lymphoid and myeloid malignancies and saying it's probably just by chance. Right? It's probably not by chance, right? It's probably germline. That's why we were asking for the skin biopsy. But, this, again, not too long ago, right? This is within days of each other, right? That I get emails like this. Molecular data are generated that we are responsible for. We really need to think about what the molecular data mean. So this is what I'm talking about. Donors have germline variants. Every human has a germline variant. Not all variants are deleterious. We have lots of benign polymorphisms out there. But we all have deleterious germline variants, even when we're not related to the patient. So here we have a patient with leukemia. Germline testing, tumor-based testing was done. We see the D140 frame shift, DDX41, common in Northern Europeans, the associated hotspot, R525H, and the nurse in the transplant program saw this report and called and said, Lucy, you're always talking about DDX41. I think this is what you've been talking about, right? And she was absolutely correct. Think about germline predisposition when you see this combination. The nurse picked it up. She was absolutely right. This person had a germline DDX41 mutation. Now, patient goes into remission, especially with DDX41, relatively chemosensitive, which is great. And now you're going to pick a donor. In this case, the transplant team did not want to do cascade testing in the family. They got nervous. They said, we just use an unrelated donor. What could go wrong? We use the unrelated donor. And at day 30, person is 100% donor. So what does the leukemia doctor do? The leukemia doctor wants to know that the R525H, the somatic variant, is gone. They'd also like to know that the germline variant is gone, but that's just going to tell them that they're all donor, which they know already from VNTR analysis. So this patient had molecular testing of this bone marrow aspirate. Whose cells are they getting DNA from? They're getting DNA from the donor. They're 100% donor. They're doing molecular profiling of the donor. If you want to do this after transplant, the pathologist should be restricting their report to the leukemia-associated somatic variant. But they don't. They just report out what their computer is programmed to do, to spit out the molecular report. And the molecular report spits out our favorite CHEK2 I200T at 50%. Who's got this mutation? The donor. So now you've done an unrelated donor transplant. You have put in homologous recombination defective stem cells that have to now go through tons of DNA replication to recapitulate a bone marrow in this recipient. Lots of DNA replication. Stem cells are quiescent cells normally. They don't like to go through DNA replication. And now if they have a mistake, they can't really fix it very well. You've set up the patient for a donor-derived leukemia. I don't know if it's going to happen. I really hope it's not going to happen. We've seen it as quickly as 13 months after transplant. This is why I say allogeneic transplant is ethically compromised, especially when related donors are used. Relatives want to help their family members, but sometimes they don't want to know if they have a germline mutation, but we kind of push them to have that. What do we do if the only HLA matched donor, related or unrelated, is a relative with a deleterious variant? We have that situation now with a patient whose only donor option is her HLA-related brother with a BRCA2 mutation. Really? Not really. She's got 2 daughters who are teenagers, and I'm pushing the transplant team. Why can't we do a haplo donor transplant? Because the patient actually in this case is BRCA2 wild type, even though there's a mutation in the family. So super complex stuff, lots of ethics involved. I really think our pathologists should be pausing, doing this testing sequentially, not in parallel. When it's 100% donor post-transplant, restrict the report to the somatic leukemia-associated mutations. Our transplant colleagues want the analysis done yesterday, right? So this is an email, Dr. Godley, who can facilitate the sibling for the HLA typing, copying Luke, who's one of our CRAs, to see if he can request the sample to do the test stat. Well, our testing takes, at best 8 to 12 weeks. Stat, like 7 to 8 weeks. That's not stat in their mind, right? It's not a stat kind of thing. So the trouble that we have are patients in remission being referred for transplant because generally the outside physicians have not done this germline testing along the way. People have trouble understanding somatic mosaicism. Clonal hematopoiesis is only somatic mosaicism. the difference with somatic reversion, this idea that hematopoietic tissues can fix themselves pretty easily, and that you get false negatives if you do germline testing on hematopoietic tissue, the importance of testing non-hematopoietic tissue because how easily hematopoietic tissue fixes itself. There's inconsistent variant classification. So our pathology department calls that check 2, my favorite allele, as uncertain significance. I don't think it's uncertain at all, and neither does ClinVar really. The vast majority of labs call this a likely pathogenic or pathogenic variant. You can see our mouse work definitely suggests this is pathogenic, and hopefully we'll publish that soon and everybody will come on board. This is a deleterious variant. So what have we learned about inherited predisposition? It's common for myeloid, and I would tell you the lymphoid malignancies even more so. Testing is very complicated. Be very careful how you interpret tumor-based and all molecular data. Prioritize patients for germline predisposition testing. The future, I think, will standardize who gets this. I would argue, but obviously I'm biased, all patients, all donors, related or not, are gonna have germline testing in the future. And I think our treatment plans maybe could get even better based on these susceptibilities conferred by these germline variants. And with that, I'm really happy for your attention tonight and happy to take your questions and comments.
Hello. Yeah.
Yes.
Yeah. Thank you, Dr. Lucy, for the beautiful presentation. So I have one quick question. So because I start the discussion, and
Yes.
please, I request to all the attendees, please use the hand raise sign so that I can identify and then ask the question. So you use the word donor-driven leukemia. So can you just elaborate this terminology? Because some of the patients we see that post-transplant, they are getting relapse. So do you feel that some of this particular group of patients get this problem of donor-driven leukemia?
Yes, when I say donor-derived leukemia, I mean it's not a relapse of the person's original disease. I mean the leukemia is coming from the transplanted cells. And now with molecular testing where you can distinguish the patient from the donor, you can tell that the new malignancy is actually from the donor. And we see this in 2 different scenarios. One, germline risk alleles in the donor that typically were not recognized at the time the donor was used, like the case that I gave to you, or clonal hematopoiesis from the donor. Those cells are predisposed to going to myeloid differentiation. And because of that pressure, I think, of the DNA replication and the prior probable damage to the bone marrow microenvironment in the recipient, you put those stem cells in this damaged microenvironment, ask them to go through DNA replication, which they don't like to do. They're quiescent cells normally. So you force them to do something they're not really that good at. You get leukemia down the road. The fastest we've seen a donor-derived leukemia, 13 months, was actually from clonal hematopoiesis. The longest I've seen donor-derived leukemia from clonal hematopoiesis was 13 years. So our patients survive a long, long time now after transplant. And we have to be very careful about complications. Now, you could argue to me, well, that person would have died a long time ago. You gave him 13 years of life. True, I get it, right? Or what do we do with my patient now who's BRCA2 wild type and her only donor is BRCA2 mutated? Again, is that really her only donor? I'm pushing a little bit here because I don't know. Should we be using the BRCA2 because it's her sibling and that's, traditionally the best donor? And maybe we give her however many years. Maybe that's more than if I don't do transplant at all. these are such difficult questions, right? And you're just weighing the risk and you don't have the crystal ball to know what's going to happen in the future. So we don't have the right answer. And I think that's okay to not have the right answer. I think what's really important is to talk to the patient and say, this is what we know. This is what we don't know. These are the risks if we do this. These are the risks if we do that. Which risk do you want to take? And just be very, very clear what the risks are. We are not testing our donors routinely for clonal hematopoiesis. We are not testing our donors routinely for germline predisposition. And my prediction is that will change over time. Now there's data that some clonal hematopoiesis is actually good from the donor, right? From DNMT3A mutated clonal hematopoiesis gives an inflammatory environment that actually helps the graft versus leukemia effect. So this can go in both directions, right? I mean, we just need much more data, but we have all the tools molecularly to be able to, know what we're doing and at least do it with our eyes open.
Yeah, thank you. Dr. Aditya, your question, please.
Good morning, Dr. Lucy. Thanks a lot for the interesting talk. So at the outset, you mentioned that the probability of finding a germline predisposition in a young MDS is close to 20%. So what is the probability of finding a germline mutation in a young MDS patient who doesn't have personal or family history of any cytopenias or malignancy, who doesn't have any physical features like early graying of hair or other physical features suggesting a germline predisposition, and there are no features of immunodeficiency. So just an MDS in a young patient, what is the clinical probability or pre-test probability of finding a germline predisposition?
Yeah, thank you for that question. What you're bringing up is really, really important. Traditionally, we were told to think about the germline when you have a strong personal family, physical stigmata, and as we peel back and require fewer and fewer, criteria, it doesn't matter. You don't need a family history. You don't need physical stigmata. Again, all of those clinical definitions were kind of positive reinforcement. As you go broader and broader, you still see it. So if you only have the diagnosis of MDS, nothing else, just the diagnosis. If you look across the age range, the answer is about 20%. Now, if you take our CIBMTR data, that's the entire age range of life. MDS from 11 years old, I think the oldest was 81. The answer is 7%. But remember, that's an underestimate because we called germline when the patient and the relative had the variant. Half the time, the donor is going to be wild type, right? When they did this transplant, they didn't know what they were doing, right? They just took the related donor. So if you multiply 7 by 2, you get 15%, right? I mean, you're right in the range of 15, 20%. So just having a young age of diagnosis for MDS is all you need. And I would argue that when we publish our paper, even 7%, every MDS patient deserves germline testing, any age, because those 7% are not concentrated in the young. They are spread across the entire age range of life. And the American College of Medical Genetics and Genomics suggests germline testing if your pretest probability is over 5%. So knowing that 7% is an underestimate, it's already over the 5%. So this study tells us every MDS. Now people will push me a little bit and say your study was only MDS patients who went to transplant, right? Because they had to have the donor. So we still need more work. We need every MDS of every age range. And you in India and the other countries there were not well represented in our study, right? Our study was mostly European centers, et cetera. So I don't know about your patient populations. You have a different baseline genetic background, right? So you all should be doing the study in your patient population. Maybe yours is higher, maybe lower, maybe exactly the same. Maybe it's the same all across the world. But for us, I do think as soon as we publish it, I'm going to push our center. Every MDS patient deserves it. I can tell you other centers in the United States are already doing germline testing for myeloid malignancy of any type diagnosed under 50. These are all arbitrary cutoffs, right? In the United States, a lot is dictated, sadly, by insurance coverage. If insurance would pay for everybody, we should just be doing everybody, right? We just haven't tried it yet because we're a little nervous that we'll get pushback, right? But I think you're absolutely right. We really don't need family history anymore. We really don't need physical stigmata. It's just the diagnosis. Now, if you have a super strong family history, don't ignore it. If you have physical stigmata, don't ignore it, but you don't need it. Just the diagnosis of a heme malignancy, in my opinion, very soon it's going to be enough. For MDS, I think it's already enough.
Thank you.
Yeah.
Dr. Harsh.
Good morning, Dr. Godley.
Hi,
Thank you very much. Thank you. Thank you very much for such a wonderful presentation and opening our eyes with regards to this CHEK2 I200T variant. My question is, do we know the frequency of this CHEK2 I200T variant in general population? And second, do you think all those individuals who have got CHEK2 I200T variant will eventually develop into MDS or not?
Yeah, that's a great question. So the general population frequency, I'd have to look on NOMAD. I can answer a related question. There are certain populations where it's higher. So for example, the Eastern European population is the highest. It's on the, say, 1 to 5% range. It's pretty high, which is why people have argued, oh, it's not contributing. It's just there in the background, right? And here in Chicago, we have the 3rd largest Polish city in the world. So people could say to me, the only reason why you see CHEK2, you have a lot of Poles, right? But this is why we went to Utah, where they don't have a lot of Polish people. They don't have a lot of Ashkenazi Jewish people. They have a tremendous genealogy tree in Utah because of the Mormon population. So they can track these families back 7, 8 generations. And we asked them, look at CHEK2. And so it was fun. The first time I talked to the genetic counselor there, she came on the phone. She said, Lucy, there's something about CHEK2 and blood cancers. I said, you don't say, you know. So even a totally different population, they see it. Now our Finnish colleagues in Finland, now they have statistical significance as well. They're actually the first people that looked at CHEK2. So I'm hoping that when we submit our paper, we have population from Chicago, we have population from Utah, we show the same phenomenon across different population. It'll be convincing to the reviewers. But please look in the Indian and other, East Asian countries, because the more we show this as a worldwide phenomenon, the more important it is. Like DDX41, we show it all across the world, right? It's all countries. And so it's very, very important that all of you look in your populations and, we see whether this is a worldwide phenomenon or coming from particular populations. And then you asked about penetrance. What's the penetrance? The penetrance for CHEK2, we actually have not formally calculated. We could do it probably, and the reviewers may push us to do it. It's not 100%. The only germline predisposition disorder that I know that's close to 100% is 5' N CEBP alpha mutations. That is so highly penetrant, you're guaranteed pretty much of getting myeloid malignancy. So there, I think, is the strongest argument for a preemptive allogeneic stem cell transplant. CHEK2, I haven't calculated, so I don't want to guess, but it's not 100%. So it's a risk factor. So we tell this to people, right? This is not a guarantee. This is a risk factor, and we just need to monitor you very closely, do our extra cancer surveillance, and my hope is you're going to be fine. Like the case I shared with you, that first pedigree, yes, we had an early breast cancer, but it's surgically curable, right? thyroid, easy to take out and replace, right? So again, it feels like whack-a-mole, right? We keep, diagnosing more and more in our patients, but at least at a stage that is very curable. That's the hope, at least.
Okay, thank you. One more question with regards to, I'm the pediatric hematologist and oncologist. With regards to pediatric leukemias, especially you already mentioned that lymphoid malignancies usually underlying the CARD11 and other all inborn errors of immunity causing genes. And do you think, do we need to do germline predisposition at the time of diagnosis, or can we do on the in remission from the hematopoietic stem cells?
Yeah, that's a really tough one, because there's the theory and the practice, right? So the theory is no. I would tell you, don't do germline from hematopoietic tissue.
Okay.
Then there's the practicality of it, right? And so I would say, in the practice, a little bit, it depends on the immediate circumstances. So sometimes we do testing on the donor from the blood, buccal swab, saliva, it's all blood, right? Sometimes we do it from the blood knowing that we can have false negatives. We always have the skin cooking in the background. So depending on the clinical scenario, it's just all about explaining the risk to the family and the patient. Right now, we're trying to be very, very comprehensive within our programs. And at least for our leukemia patients coming to transplant, 95% who should have had germline testing have had it at the time they get referred to transplant. Again, the problem is these people coming from the outside in remission being referred just for transplant, because then you want the transplant yesterday and there's not time to do the testing. So I'm a bit of a purist. I get really nervous about the hematopoietic reversion, especially in kids, because the SAMD9, D9L, that's where you see it in the kids. We've actually never diagnosed it in the adults. So it makes me very nervous to use tissue that's hematopoietic, even in remission, because of this issue of correction. And that's a real phenomenon there. So, I'm very, very cautious about doing that.
Thank you. Thank you very much.
Mm-hmm.
Yeah, Dr. Jasmita, please. I think we're muted.
Thank you for this excellent talk that you took, and my congratulations on the design of the bioinformatics pipeline and the assay that you designed. It's not really something that we see on a regular basis being done. So with respect to, so you said that with the somatic testing again, so if something is at over 25% level, you would like to monitor your patient subsequently, and if the mutation rate stays stable, those are the patients in whom you would consider germline testing. So for that, do you use the same assay, or do you use the regular somatic assay, say for genes that are covered in the targeted sequencing panels that our patients usually undergo?
Yeah, thank you for that. Thank you for that question. So actually, we only need to see the variant on the tumor-based profiling once to prioritize that patient for proper germline testing. The lowest variant allele frequency we have seen on that testing platform that has been germline is 31%. That was a BRCA1 mutation. And that's why right now our VAF cutoff is 25%. It's a bit lower than the lowest number we've ever seen. I hold out that that number could go lower because there's, technical variability over time. And so what we do is I look at every variant called from that platform. Actually, I just got the data yesterday for May. So once a month, I get a computer dump of the data and I look with my lens. And I talk to the clinic doctor of the person. I say, your patient was found to have this variant at this VAF. This could be germline. Do you want us to get involved? Sometimes they want us to call the patient. Sometimes they want to talk to the patient. But in any case, we do the skin biopsy and we send the skin biopsy to SomaDas Human Genetics, the proper germline test. I think it's very important. Again, I'm a bit of a purist, That pathology molecular tumor-based panel was designed to find somatic tumor-associated variants. It was not designed to be a germline test. So I don't ask them to do what they didn't design it for. If you see something there, don't ignore it, right? So when we see something on that test, then we bring the patient to what I consider the proper test. Again, the second way we do this is if you can document that variant in a relative, you know it's germline. The problem is you can go through a bunch of relatives and they're all negative because by chance they don't have it. So it's only helpful if you see it in a relative. But if you have like a brother who's also had cancer or the mother's had 4 cancers, sometimes it's pretty obvious which family member to test. Sometimes you're surprised, like my patient who's negative for the BRCA2 mutation, right? We did testing on her, by the way, with a larger panel. It's not like there's a second mutation. She's negative. So it's just some environmental thing or some gene we don't know of yet, right? So sometimes you can be surprised, but again, we define germline to be a variant at a germline associated VAF from skin fibroblast, or the variant is in a relative.
Yeah. Thank you.
Yeah, I will take a question from the chat box. So Dr. Raj has a question. So is this more important? I think he wanted to mean the germ cell mutation analysis for transplant for benign diseases like thalassemia or sickle cell disease, etc.
This is a great question. What is the frequency of germline mutation? Or sorry, what's the frequency of deleterious germline variants that can predispose to these diseases in people with other germline conditions associated with benign hemoglobinopathy? We call them benign hemoglobinopathies. So first of all, sickle cell disease is associated with baseline inflammation that is associated with a leukemia risk. So there are 2 studies that show baseline inflammation, a higher level of diagnosis of leukemia in sickle cell patients, not present in thalassemia patients probably. So these patient populations should not be thought of completely the same. They aren't. There is a baseline inflammation in sickle cell disease, not in thalassemia, that already gives sickle cell patients a higher probability. Definitely these individuals can have germline predisposition like anybody else. And so I think you need to look again at the personal family history of these families. Should we be doing germline testing on anybody with a hemoglobinopathy having a gene therapy type transplant right now, or any allogeneic transplant right now? I think you know how I would answer that question. Yes, you should think about it. I know of several cases where there has been leukemia after these kind of transplants, and the individual not only had the germline hemoglobinopathy, also had germline predisposition to leukemia. So it's all out there, right? And the more you think about it, the more you test for it, the more you're going to see it.
Thank you. So there is another question, that will you pursue a transplant in cases of donor-derived chip?
This is a great question also. And this is what the transplant community is grappling with. You have data now coming out that some clonal hematopoiesis is good for transplant because there's a low-level inflammation that stimulates the T cells to find the malignant cells. But too much of a good thing is not good, right? And other inflammatory associated chip is not good because it gives too much graft versus host disease. Who knows where it's really going to all fall out, right? So I don't, it's a long conversation with patients, right? If you have history, personal family history, suggestive of germline predisposition, consider testing before transplant, consider testing donors for clonal hematopoiesis, and then when you get the result, be ready to have a long conversation with the patient. Which mutation is it? You know, could it be associated with a better outcome? Could it be associated with more graft versus host disease, donor-derived leukemia afterwards? we don't have all the data. And actually, I find that patients are more comfortable with the idea that we don't have all the data than our colleagues. There is an expectation in medicine that we know everything. And I think colleagues are nervous to tell patients we don't know everything. But I think patients understand that we don't know everything. So I have no problem telling the patient, this is where I have my knowledge to this point today. This is what we don't know yet. And we all have to do what we can with the knowledge that we have at this moment, because this is the moment we have to make a decision. Maybe in the future, we would have made a different decision, but we didn't have the knowledge at that time, right? So I think if you're just very straightforward with people, what we know, what we don't know, what is the risk that we're taking if we do this or that, then we can make a decision together with our patients. We can all feel like, okay, we made the best decision we could. The patient chose whatever road we took for these reasons, and we're all comfortable moving forward.
Thank you. Dr. Mukul?
Thank you, Dr. Lucy, and a very good talk. So it's an idea from many of the things. So we are mostly testing with the blood samples or the marrow samples only as a practice. So problem is that we do get variants, maybe sometimes, let's say, in a DNA repair mechanism genes, like FANC and other things. So do you modify treatment? Suppose we understand that FANC they do have increased sensitivity to chemotherapy. But these patients who are accidentally picked, say late in their 20s or 30s and no other phenotypic features or other features with a MDS or a leukemia, barring FANC, do you recommend the changing of therapy, chemotherapy, especially for these patients?
Yeah, it's a great question. Generally, the only patients we modify the treatment for, as you say, are Fanconi anemia or telomere biology disorder patients. Sorry, it's morning time here. People are mowing their lawns. Maybe can you hear me okay, or should I close the window?
Very well, very well.
Okay, okay. So generally, actually, the people with germline predisposition have relatively chemosensitive diseases. So we treat them according to, the regular protocols. I wonder in the future, though, if that will change. For example, the homologous recombination DNA repair should we be using PARP inhibitors for those patients, right? Just like for BRCA1 and 2 solid tumors, or even preventive strategies in the future. We don't do any of that today. We treat most of our patients exactly the same way. The only modification is thinking about the relative at transplant very carefully. And if at all possible, using the HLA matched donor who does not have the mutation, if it's possible to be done.
And this mutation is to be tested only and only from the skin fibroblast?
For me, yes. Or the relative has the mutation. So you can define germline because the patient has that variant at a high VAF in the skin fibroblast, or the relative has the same mutation. So the sister, the son, the brother, the mother, so we define germline with family segregation or in the skin. So sometimes family segregation is easier for whatever reason, if the patient's not going to have a bone marrow biopsy or they're really tired of having procedures, or you need to do it really fast. So sometimes we bring the whole family into the clinic, we draw blood on everybody and just do it, with the caveat that blood is not perfect, but we need this result really fast. And then we have the skin, we do the skin biopsy, but it's cooking in the background to show that it was really, often the family segregation is very helpful because often somebody else in the family has it.
I have a bad feeling or a hunch that the strategy might decrease the sibling and they have to transplant to a great extent. What is your take on that?
So my take, here's my question. The people who've had bad outcome. Oh, let me say one other thing that I have not had time to talk to you about yet. most of the time when we do a transplant, we tell people, the germline patients, all we need to do is bring in wild type stem cells. You're going to be fine. RUNX1, CEBP alpha, you bring in the wild type stem cell, perfect, right? They're perfect for life. Well, we started noticing at the University of Chicago, this is not true for DDX41. You bring in the wild type stem cell and they were dying of graft versus host disease, dying in 2022 of GVH, like severe GVH that you cannot control. So then I start calling around to my friends around the country, our colleagues in Australia with whom we work really closely. What's happening to your DDX41 patients? And some of them were saying they've got GVH, and others were saying they do just fine. So then we start asking, when they have the GVH, what kind of transplant do you do? What kind of GVH prophylaxis do you use? What we found is that the centers that were using post-transplant Cytoxan, no GVH. Our center and other centers that don't use post-transplant Cytoxan, rip-roaring GVH. So what I think is the DDX41, remember it's part of the STING pathway, innate immunity pathway. I think all the organs that have the DDX41, something is presenting something, some inflammatory something to these T cells coming in, right? And these T cells are going crazy and killing our patients. And somehow the post-transplant Cytoxan is able to ameliorate it. So this is another paper that we have under review right now. We're just answering the reviewer comments now. I hope, because I really think this has an important clinical impact. I have a patient, DDX41, coming to transplant, and I'm gonna go crazy. The transplant team has to use post-transplant Cytoxan this time. Now, a lot of centers that use post-transplant Cytoxan, they think everybody should get it anyway. Maybe that's true, but definitely the patients with germline DDX41. So that's one germline syndrome where I do think the treatment should be different. If you're going to do a transplant DDX41, those recipients have to get post-transplant Cytoxan. So I want to go back in time and look at all the severe GVH across the centers and genotype them for DDX41. How many times were they DDX41? You know, like I was talking to Fred Applebaum at Fred Hutch, and he said, oh, our patients do super well with transplant, super well. I said, well, what's your percent of grade 3, 4 GVH? Only 5%. I said, that's your DDX41s. Go genotype them. Right? So that would be really cool, right? If we all went back to our transplant centers, who were the people with severe GVH? Is the germline DDX41 preferentially found there? Especially if you don't use post-transplant Cytoxan. That's my prediction. So I would say besides the telomere biologies, besides the Fanconis, the DDX41s need a special treatment at transplant. So thank you for your question. I didn't have time to talk about that.
Dr. Lucy, I have one quick question. So many times we see in a routine checkup, a routine health checkup, patient comes with a normal CBC with a macrocytosis of say 100 and 102 MCV.
Right.
Hemoglobin is damn good, 13, 14. They are not vitamin B12 deficient. Right. They are not alcoholic. Clinically, there is no indication of doing the bone marrow aspiration biopsy. Yeah. Hemoglobin good, WBC good, differential good, platelet good. Yeah. So what is your comment on that? You should do these patients particularly because they ask why these parameters is abnormal.
Exactly. I have a whole bunch of these patients because the primary care doctors rightfully send them. I mean, I get it. And you do the B12 and you do the folate and da-da-da-da-da. I do the bone marrow biopsy, I do chromosomes, I do molecular, bland, and I just follow the people. Because there's something going on, right? I mean, I don't know. I mean, probably there are some people that just have a germline macrocytosis that's nothing bad, but it just makes you worried that something is cooking, right? So I do a whole workup, very expensive workup, And then I just follow them clinically with the blood counts. And I have one lady, her MCV is going up, 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 up every time. But there's nothing to treat. There's no dysplasia, no molecular, no chromosomes. So I just watch.
Yeah, Dr. Mukul, please.
Ma'am, you were very specific about MDS EB2 in the young patients. We in India, as a belief, and almost all my colleagues will agree that our patient age with myeloid malignancies is very much low. So we do get a lot of pediatric, adolescent, young adult malignancies. So with respect to, let's say, pediatric or adolescent AML or CML, what percentage of germline mutations are you expecting in these patients?
Yeah, these are such great questions. So for all AML, no one's really done the study. So for all AML, people have looked for DDX41 because it's the most common germline predisposition disorder for adults AML. The answer is about 1 to 2% of all AML is from germline DDX41. Now we need to do the studies, right? Take all myeloid malignancies across the entire age range, germline on everybody, what's the percentage? These studies will be done. Right now, what we started with was getting a percentage that people would pay attention to. So 20% young MDS, now broaden all MDS, now broaden to all myeloid malignancies. So I don't have the data today, but it's detectable. I mean, if 1 or 2% of all AML is DDX41, you know that the answer is higher than that for all the disorders. So this is why I say, I think very soon, everybody's going to get germline testing. It's just that prior, nobody looked. So if you don't look, you're not going to find it. If you don't look, you're not going to diagnose it, right? Now you change your lens and it's everywhere. How did the field miss it? I mean, it's not hard to find it. I don't know. It's kind of blows my mind that people looked for the somatic without thinking about the germline all these years.
Thank you, Dr. Lucy. Any questions from the discussants?
Yeah, I can maybe answer some of these quickly.
Yeah.
Are there data on how many donors develop malignancy after donation? Was there simultaneous leukemia in the recipient? The answer is yes to both. This has been documented in the literature, especially for germline RUNX1 and CEBP alpha. So I would say it's in the United States, at least, absolute malpractice to use a germline mutated RUNX1 or CEBP alpha donor. It is not clear about BRCA1 or 2. If the donor has those, is it bad? Are there some germline mutations that are permissive? We don't have enough information. We thought that our CIBMTR study would give us the information because we now know which donors were mutated. What were the outcomes of the people, the MDS patients, when the relative had the germline mutation? The problem actually is when you look at the genes of the 7% positive, it's a smattering across like 30 different genes. So we don't have the power. We only have 3 DDX41s, 2 BRCA1. It's not a powerful enough study, even though it was 400 pairs. We need a much larger study to be able to answer the question. Do I think patients with solid tumors will get germline testing? In the United States, they already do. The solid tumor doctors were way ahead of the heme doctors on germline predisposition testing. I don't think there's any difference mechanistically between solid tumors and blood cancers. They're all cells dividing. It's all DNA repair on the adult side, all telomere biology. So there's really no difference. The syndromes all overlap. Data on possibilities of clonal evolution in recipients receiving transplants. VUS, so VUS's variants of uncertain significance, we don't follow clinically. We follow them on a research basis and try to push them into pathogenic range. But if I can extend the question to be about clonal evolution or clonal hematopoiesis, what's the frequency of clonal hematopoiesis in these disorders? Very high for RUNX1 and GATA2. Not so high in DDX41. caveat here is the average age of diagnosis of the malignancy in DDX41 is decades later. So is the presence of clonal hematopoiesis a molecular clock? As soon as you have the clonal hematopoiesis, you're 10, 15, 20 years away from getting your malignancy. That's possible. And so as our DDX41 mutation carriers get a little bit older, we might start seeing the clonal hematopoiesis. Now the question is, if you see clones growing, like a B core mutated clone growing in somebody with germline RUNX1, is that molecular MDS? Is that enough for you to take that patient to transplant? Depends on who you ask, right? Some people feel strongly yes, others feel very strongly no. So I think we just need more data. Is the germline panel covering the same genes as the somatic panel? No. The germline is going to cover SAMD9, D9L. We don't see those on our somatic panel. You could imagine a very, very large somatic panel that covered all the germline predisposition, but remember the somatic panel is being done on hematopoietic tissue. Therefore, it should never, in my opinion, be used as a proper germline test because of somatic reversion in the hematopoietic tissue. You will always have false negatives if you're using hematopoietic tissue to look at germline.
Dr. Mukul, do you have any question?
Yeah, very small question. So, with respect to targeted molecules which are currently coming up and more are likely to come up in the future, let's say we have a targeted molecule again, TP53, API246. So, do we expect better responses or worse responses in somatic or germline mutation in such patients?
So we don't really know. For Li-Fraumeni, we don't really know. For DDX41, they're generally chemosensitive. BRCA1 and 2, generally chemosensitive. RUNX1, my experience, for someone who had a true 5q minus syndrome with germline RUNX1, exquisitely sensitive to lenalidomide, went into complete remission within a month. So as I said before, generally we treat the malignancy developing in these people exactly the same as you would otherwise. The difference is at first of all, I think these people have to go to transplant. And whenever possible, using a wild type donor, again, for DDX41, then with post-transplant Cytoxan. You know, this is even controversial. My opinion is still controversial. Take, for example, germline CEBP alpha. Some would say that's so exquisitely chemosensitive that you should wait for them to get relapse, which I put in quotes, before you transplant. I put the relapse in quotes because Jude Fitzgibbon looked molecularly at the acquired mutation. That's a molecular fingerprint for the somatic mutation. And when they relapse, quote unquote, it's a second leukemia. That 3' mutation is not what it was originally. So because you still have the germline mutation, you still are at risk for developing second, third leukemias down the road. So again, have a conversation with your patient. If you have a first remission, 5' N CEBP alpha germline, you can tell them, look, we have a choice. We take you to transplant now, or we can wait years. And if you develop a second malignancy, take you to transplant then. And have the patient decide. My personal preference, if I were the patient, I would go to transplant younger. Right? I'd go to transplant at first remission. Why would I wait to get worse GVH? And maybe I wouldn't get into remission the next time. So that's my opinion. But when I said that at MD Anderson, trust me, if they could have thrown rotten tomatoes at me, they were just vile in their response, like completely thought I was crazy. So people have their opinions very strongly, you know. And I think in the end, we just have to share the data with the patient and help them pick what risk they want.
So thank you, Dr. Lucy, for your lovely
Sure. Well,
presentation.
thank you all. It's very late for you. I feel bad.
No worries at all. This is a rare opportunity to learn such a beautiful thing from expert like you. And I'm sure it will change a lot of our perspective in the coming days into practice. Wonderful lecture, madam, and wonderful discussion. So
thank you so much. And thanks again for having me. I hope we can see each other in person at some point.
You already came to Mumbai once and we'll call you soon.
Exactly.
When everything opens up perfectly. Thank you, Natco, for this lovely platform and the Perfect Square for the fantastic logistics. And
Yeah, it
thanks
was really
all
great.
the discussions, Dr. Mukul, Dr. Aditya, Dr. Harsha, Dr. Jasmita. Thanks a lot for joining this beautiful meeting and the good discussion ahead. Thanks a lot.
Thanks, everybody. Have a good night.
A good night.
Bye-bye.
Bye.